Good morning, everyone. Welcome to the 23rd Annual College of Arts and Sciences Spring Symposium. My name is Amy Kierstead. I'm an Associate Dean here in the College of Arts and Sciences, and I'm here with my friend and colleague, Dean Jonathan Millen.
Good morning, everybody. Good morning, Amy.
Good morning, Jonathan. You know, this is our 23rd Annual Symposium and a showcase of student research, scholarship, and creative activity, and We've got 60 some odd posters this morning, 130 students packed here in this gym, and we're super excited about this. We've got oral presentations this afternoon. We have one of our alumni coming to talk to our students over lunch. It is an action-packed day.
It is, and what better action than Facebook Live, as I understand it. In addition to the work of the students, what we're going to see is also the work of our faculty who have mentored these students in the classroom. Some of these projects come from classroom-based assignments, students working in the field doing their research, and of course students in the lab conducting their experiments. And what you end up with is a broad spectrum across the arts and sciences of engaged students doing top-notch scholarship and research. And we're very excited to share a small sample of that with you. But to get the whole picture, come on down to campus. We're in Biddeford, we're in the campus center. There's hundreds of posters, hundreds of folks. What do you say we start with the fun?
Let's, pun fully intended, let's dive right in.
Let's do it.
All right. Okay, so we are going to come over here to our first project. Here we have Ray Pesic, who is a first-year student studying medical biology, and Sarah Swanek, who is a graduating senior studying medical biology. Did I say marine biology? I meant medical biology. And they are working in the research lab of Dr. Kristen Burkholder and in collaboration with Dr. Carrie Byron. And they're going to tell us about microplastic contamination and tissue distribution in Atlantic sea scallops. All right, Ray, take it away.
Okay, so you might be wondering what a microplastic is. It's defined as a plastic that's smaller than 5 millimeters in size. Although it usually ranges from 1 micron to 5 millimeters, and it's usually closer to the 1 micron size because you usually cannot see it with the naked eye. Microplastics are a huge aquatic pollutant. You find them in everything you use. You can find them in makeup. You find them in all of your clothes. So many. Textile waste is the biggest contributor to microplastics. So this is a huge aquatic pollutant, and you can find it a lot in filter feeding bivalves. So we're going to look at scallops. That's what our project's looking at. These are from 5 years ago. They were freeze-dried. But this is the digestive tract in the scallop, and this is the part that we eat. So this digestive tract is where all of the microplastics are getting trapped when they first take in all of their nutrients to eat. And then there's studies that have shown that there's translocation in fish from their bloodstream, how they eat, to then the parts that we eat. So we're looking at filter feeding bivalves, specifically the scallops, to see the plastic contamination in the viscera and then into the muscle, because the muscle is the part that we eat, and that's concerning if we're ingesting it into our body. So there's also depuration, which is when you put these scallops from the ocean into fresh water, clean out all the sludge and everything. And that doesn't get rid of microplastics either. It's not designed to get rid of microplastics. And so we're still left with microplastics, and so our study is designed to look at what's in the muscle versus the viscera.
So to do this, we take our scallop samples. We have freeze-dried scallop samples from Villenhaven, Maine. So you can see the muscle versus the viscera. It does look different. We pulverize these samples into a powder, and then we put them in hydrogen peroxide. They stay in the incubator at a higher temperature for a couple days, and then we put the sludge through a filter series. So the first step is through glass wool. That gets out the larger chunks that wouldn't image anyways, and then we use a vacuum filter on a 1.2 micron filter, and we take this filter and image it. We use a slide scanner, which is basically a very fancy microscope that takes a picture of the whole slide instead of just one section. And then we have to refocus it onto the microplastic layer. It usually focuses on the filter layer instead of the microplastic layer, so we have to usually bring it back every time. And then we take a 25 picture stack of whatever we're trying to focus on. Then we can compress that image and darken it. We upload it to ImageJ, and we count the fibers. So you can see the fibers, they look like fibers with a darker outline here. We can count those in the muscle versus the viscera. We put them on a graph here to easily see. There's a lot more microplastics in the viscera, which is what we'd expect, because that's the part that's filtering out any type of waste. But we also are seeing them in the muscle. It's less, but it's still there, which was the point, was to see if it's still there or not.
This is fascinating. Thank you both so much. What a great way to get our morning started. The science is really fascinating. It really is. But what I like about this project is the real world applicability is so obvious. So I'm going to cut right to the chase. I love scallops. I love my seafood. Should I be nervous? Should I continue to eat when I go out? Or how do I take this information and adjust my own life?
I mean, I think that it's still going to be in everything you eat. Scallops are a better choice compared to oysters. We eat the whole digestive tract of oysters, so they're a better choice comparatively. I will still eat scallops because,
I feel better already. Thank you.
Again, it's still like it's a world problem. It's not necessarily an individual problem because it's still in everything that we eat. It's still going to be in these other foods that we eat. So it's not like you're necessarily cutting it out of your diet.
That's a great segue because I want to ask you a question. I just learned about your plans for next year. Why don't you tell us?
I'm starting at University of New England's College of Osteopathic Medicine in July.
And so then if it's a world problem, if it's a health issue, as a future doctor, how do you make sense of that? Will you educate your patients differently? Does it influence the way you think you'd practice medicine?
One of the things that I think is really interesting about our project, it's an offshoot of a larger project. It's actually the poster next to me, but they're looking at the ability of microbes to colonize in marine organisms specific to microplastics. So if bacteria can bind to these microplastics, they travel a lot faster in the ocean than the bacteria would just in their colony form or if they're in other organisms. So if those are pathogenic bacteria, could that then spread disease a lot faster? Could you be seeing different types of infections from shellfish, from what you're eating? That would be my area of interest, is seeing more of like a disease process of, is this a problem that's going to make disease spread faster?
Well, I feel safer already, and I'm hungry as well, so I hope that's not a contradiction in terms. You have kicked off our day on such a great note. This is wonderful. All the best to you. We'll look forward to seeing you on campus, and you're a first-year student, so we'll see you as well. Fantastic. Thank you both so much.
Thank you to you both.
Amy, what a great start. This is amazing.
I know. I told you we were going to dive right in. Jonathan, one of the things that I love about being in our College of Arts and Sciences are all the different ways that students can engage with undergraduate research. So, a lot of students will start in a similar way that Ray did, which is volunteering. Some students will use their federal work study and have employment by working in a research lab or engaging in that way. But they eventually move to earning course credit. And so what a way to build up those course credits and get that hands-on experience.
Absolutely. And I don't know if I made the point, but we had a first-year student and a fourth-year student working together on the same project.
Absolutely.
I think that's rare.
Well, and so
amazing.
many mentoring opportunities and places to grow. But one of the things about Ray is that she's going to continue this project through the summer as part of our SURE program.
The SURE program is our
summer undergraduate research experience.
How's that for a softball?
Well, I was going to say, why don't you tell us a little more about that? This isn't scripted at all. So that's where our students stay here on campus, working under the direction of a faculty mentor and engage full-time with those research projects.
Maybe we do a script next year. I don't know.
Maybe we should,
maybe not. We'll see.
But one of the great things about that is now we generate this community of scholars where they can really dedicate their time, get that field work in, and it's a really fun experience.
We'll have
for
about
students.
25 some odd students on campus.
Absolutely, we'll have more than that, just through all the different ways that students engage. But it's always a great time. So, and speaking of SURE students, our next student was one of our SURE students and also engaged with research for course credit. Here we have Sarah Padalero. Sarah is graduating with her Bachelor of Science in Biochemistry, and she's been working on a really neat project under the direction of Dr. Amy DeVue in our School of Mathematical and Physical Sciences and in collaboration with Dr. Dina Small from the same school. And so she's going to tell us about structural characterization and toxicology analysis of polyphenolic derivatives with antimicrobial properties. I'm the chemist, so I get to read the title. All right, Sarah, welcome.
Thank you very much. I really appreciate it. So I'm Sarah Padalaro, and as Dr. Kay said, I am a senior biochemistry student here at UNE. So for the past 2 years, I've had the opportunity to conduct undergraduate research with Dr. DeVue, as well as Dina Small, and my research partner, Carolyn Curley. And so the importance of our research is right now, antimicrobial resistance is a huge issue in modern medicine. And so new compounds with structural diversity are needed to combat the global human health threat. And so what we did was, working off of previous work from graduate student Amber Cusson, we synthesized novel molecules. With this synthesis, we take the compounds that we make and we run different analyses for structural characterization. And that helps us confirm or deny and support that our products are the desired ones that we hoped to have. And so after running the structural characterization, we were able to take it a step further in our advanced biochemistry lab and run a toxicology analysis with our compound that we made, which was really cool because we were able to, step one, make the compound, and then step two, actually use it with zebrafish. And so that was a great opportunity to have. And so what we did was we did different morphological studies, behavioral analysis, as well as protein expression to see, like, what does our compound do when it's exposed to zebrafish. So yeah.
So that's fascinating. And now, Sarah, I know we have a zebrafish facility right here on campus.
Yes.
So there has to be a reason why you chose to use the zebrafish. Is it because we have them, or is there something unique about zebrafish that lends itself to this kind of analysis?
Yeah, so a part of our advanced biochemistry lab was utilizing zebrafish, so that was part of it. And just the overall husbandry of zebrafish is very efficient. And like you said, we have it right on campus, and we have people that take care of them. So when we aren't here on the weekends, we know they're being taken care of and monitored as well. And zebrafish are also a homologous species to humans, meaning that a lot of their genes are similar. And we can use the data obtained from this analysis to kind of compare, like, what would this look like in humans. So zebrafish are just an amazing species to be able to utilize, and luckily we have that here at UNE to use.
And you said you started this in a course project, in a chemistry course with Dr. DeVue. And was that sophomore year?
Yes.
And so you started, and you've been working on this then for 3 years?
Yes, yeah. So none of this would have been possible without the support of the College of Arts and Sciences, as well as the Summer Undergraduate Research Experience Award. And so that's really where it started, the summer of my sophomore year. I think it was second summer of COVID is what we sometimes refer it to. And so we were able to take the project, worked on previously by Amber Cusson and Dr. DeVue, and that's really where the spark started, and we've been working on it up until this semester. And unfortunately, me and my research partner, Carolyn, are graduating. But it was just a great experience to have, and to see the outcomes that we've been able to do has been great, and I really appreciate it.
That's awesome. Now, you need to get a shot of Sarah and I together, because on Facebook, you might have seen us before, out on Freddie Beach when we did a nice polar plunge a little while ago.
Yes, we did.
So give it up there. Sarah's great. It's a great presentation. Thank you so much for sharing it with us this morning.
Thank you very much. I appreciate the opportunity.
Thanks so much, Sarah. Dean Millen, are you ready for your chemistry quiz?
How about we move on to the next presentation?
I think that's a great idea.
Yeah, I can give you a chemistry quiz, but I can't pronounce the terms the way you did, so.
That's fine.
I'm going to save some face on Facebook Live.
That sounds great. That sounds great. I'm sure Sarah told you about how that project came to evolution a little bit.
Yeah, we did talk about that.
And if we can just continue the conversation we were having moments ago about the ways to engage, those cure projects that are embedded within courses are such a great opportunity for students to have an authentic research experience without needing to register for additional credits or do additional work. So that's one way.
Yeah, it's a great example of engaged learning, active learning, where it's no longer just a professor standing in the front of the room and saying, this is what it looks like with a textbook or an overhead slide. We have our students doing the research in the class in addition to the lab.
Absolutely.
And that full integration of the curriculum is really outstanding.
Absolutely. And that's not limited to science. We have faculty in our humanities programs that may go into the archives and work with students to develop a unique museum exhibit. We've got an archaeology professor who's out on Freddie Beach and doing digs. So learning by living, if you will.
Learning by living, I love it, absolutely.
Yeah. And of course, those aren't the only ways. We also have research methods courses, we have capstone courses, we have different ways that students can continue with their curriculum and be engaged. And in fact, one of those capstone students is here with us right now. So here we have Jillian Courier, Jillian is a senior marine affairs major, and she's going to tell us about her marine affairs capstone project, the fall of the lobstering industry in Maine due to climate change. So, welcome, Jillian.
Hi. So, as Kirsten already talked about, I decided to look at the fall of the lobstering industry in Maine due to climate change because I wanted to look at the importance of the history, cultural, and economic value of lobsters in general. And I really saw that throughout my research. In regards to history, the first lobsters that were documented to be caught were done by English settlers in the 1600s, and lobsters were even being caught by hand still in the 1800s. A majority of the American lobsters are caught in the Gulf of Maine, as well as Maine specifically, and it is a $1 million industry that is very impactful for our coastal communities, as well as our coastal economy. So unfortunately, it is being impacted by climate change, and two aspects are the increase in water temperature as well as ocean acidification. The Gulf of Maine has experienced one of the fastest increasing water temperatures in any marine ecosystem, which is very problematic because lobsters like cold water. So they're going north towards Canada or further offshore. And of course, ocean acidification results in fewer carbonate ions being in the water, and a lot of shellfish are made out of calcium carbonate shells, and one of those shellfish include lobsters. And they're going to be directly impacted, and some of the effects of the climate change are shell disease as well as larval stage disruption. Shell disease is a naturally occurring bacteria that eats away at the shells of the lobsters, and luckily lobsters can get rid of the disease when they molt their shells, but the lobsters that are more at risk are the female lobsters that are breeding that have eggs, because they have to keep their shells for a longer period of time, so they can actually die if they do not molt their shells soon enough. And then larval stage disruption is a big problem because ocean acidification results in their shells taking a longer time to grow, so they are being in a more vulnerable state for a longer period of time. But there is hope for the lobstering industry. I will not get into that right now, but there is hope.
I hope there's hope because I love my lobster. We just learned about scallops earlier, and I don't want to end my day with the notion that I can't eat my seafood.
Absolutely.
But that's for another time. What really fascinates me, Jillian, about your talk is that you're looking at an issue that deals with marine science, but you're not looking at it from a scientific point of view per se.
Yes.
It's really interdisciplinary. I heard you talk about history, the economy, global effects, and the like. So can you tell me a little bit about your major? And I think most folks are familiar with marine sciences, but your program is a little bit different.
Yeah, so marine affairs is a mix between marine biology, economics, law, and education. So it's basically the social aspect of marine science. So it's more of communicating with people, and that's really what I want to do. I want to work with education outreach programs because there needs to be communication with science and all things like that, because that's super valuable.
Yeah, and I know we talked about it a little bit earlier. I know we don't have time to go into it right now, but we were talking a little bit about the whale issue, right? And the controversy right now between the lobster fishers and then the right whales and their challenges. And so is this the kind of issue that folks in marine affairs would be diving into?
Absolutely. I actually wrote a paper for my class, Science and Society, maybe 3 weeks ago about this exact debate. There's a lot going on, a lot of opinions. I could totally dive into it, but not right now. Yeah. But yeah, it definitely also interacts with kind of my research, but yeah.
And then you also mentioned your interest in communication and science.
Yes.
Tell us just quickly about some of your plans once you graduate.
Yeah, so I really want to focus in on science communication because there is this constant gap between scientists and the public, as well as scientists and fishermen, which leaves a lot of people being confused and feeling like they're not understood. And everyone has a right to have knowledge. So the public deserves to understand big concepts like climate change and what is that going to do to their lives, and what is it going to do to their lobsters, and what are we going to do with fisheries management, and just how are we going to have these really impactful, influential conversations. I want to be a part of those conversations and figuring out how to facilitate that and bridge the gap and be the interpreter between the scientists and the public.
Well, as a dean of a College of Arts and Sciences, that warms my heart, because that's exactly what we need, is the ability to take all this amazing science and knowledge that we create and to translate that to the public and have the public find that real practical value in it. I feel so much better just hearing you say that. Thank you so much.
Thank you so much.
Great. Thanks so much, Jillian. That was really exciting.
I'm already just so blown away, Amy, by what I'm learning this morning.
I know,
it's amazing.
and these are all undergraduate students.
I know it.
Freshmen all the way to seniors.
It's remarkable.
So, yeah,
Absolutely remarkable.
We always say that our undergraduates here do the work of graduate students, and yet we also have graduate programs. Here in the College of Arts and Sciences, we have traditional 2-year master's programs in biological sciences and marine sciences. And, of course, we also have our accelerated programs, our 4+1 programs.
And the 4+1 programs are for our own students?
They are for our own students. And so it's a way for students to start with their research and their graduate courses in their senior year and get the bachelor's master's program in one fewer year. And we have those in biological sciences, marine sciences, and environmental sciences.
So we have the traditional +2 program, which is usually for students that come to us as
Correct.
grad students,
Right.
and the +1 option, the 4+1 is for our own students to be able to continue in these programs.
Absolutely, absolutely. And that's a great segue, because here we have Katie McGee, who came to us from Johnson & Wales University, but she's here now, and she is working with Dr. Noah Perlett. This project was also in collaboration with Dr. Will Kachitsky, and she's going to tell us about how to determine environmental conditions along bobolink routes for their migration. So, Katie, take it away.
Thank you. Yeah, so like you said, this project is part of my master's section of the remote sensing class I took with Will. And what we're doing here is kind of developing a protocol for extracting the environmental data that I needed for my thesis. So my thesis, in a broader sense, is looking at how both genetics and environmental conditions might be influencing migration behavior in these birds. And so for this poster, I'm just focusing on the environmental conditions that we are looking for. And so pretty much what we did is we have GPS points for geolocators for these birds from 2009 all the way to 2022. 30 different birds that we put little geolocator backpacks on and followed for their entire migration path from North America, in where we collect them in Vermont, all the way down to South America. And we have all these different points so we can see everywhere it's been every day along its migration path. And what I wanted to do was to determine what were the environmental conditions that they were experiencing along their migration path. So how do you find out what a bird experienced back in 2009 when it stopped over in the Caribbean, right? And so there's these huge environmental databases that the European Union puts together. And so I went through these different databases and looked at the hourly data that for different environmental factors for pretty much its entire migration route. So I looked at 2-meter temperature, so the temperature like right above ground surface, total precipitation, mean sea level pressure, and also leaf area index, and able to overlay all of these different maps with my birds' data points. And I created this, Will helped me create this Python code that filters through these hundreds of thousands of data sets for the environmental conditions and pull out and extract exactly the temperature and exactly the different environmental conditions that bird experienced on that day. And so it starts creating these really interesting stories where we're able to look at these broader patterns across a bird's migration path. So we can look at how a migration path from north to south here, going from yellow is north, down to south, back to north again. And we're able to see these patterns emerging of these birds' behavior where temperatures plummets, and it's like, oh, I need to move farther south again. And it'll do a jump and move farther south. And this continues all the way down. So it'll hit a low temperature, move farther south, hit a low temperature, move further south, until it's all the way down in its southernmost reaches of its migration. And then it actually jumps, you can see here, this particular bird hit a really cold day all the way down in South America. And you can see that's when it starts its migration north again. And so in the matter of a couple of weeks, this bird travels from southern South America all the way back up to North America again. And we can start piecing together this really interesting story.
Yeah, and that's just fascinating because put your hand back up there, would you, for a second? Just in the shot.
Yeah.
So look at the bird. So that's about life science. With your hand in the shot.
That's so real.
and what you're telling me is that little bird is flying from Vermont,
yes,
in New England,
to
the bottom tip of South America.
Bottom tip of South America.
That's not possible.
They're very small. It is possible. They're brilliant birds. They're so strong. And yeah, these little geolocators that we put on them are teeny tiny. They wear them on as almost like a backpack. It wraps around their wings, and they're fully able to move their wings. And they carry these little monitors for us, all the way down, so that we can see where they're going.
Now here's what everybody wants to know.
Yes.
It's not so much how do you get the little backpack on the bird. I'm sure you have a way of doing that. But how do you capture the bird in the first place?
That's such a good question.
Oh, thank you.
So we use things called mist nets, and they're a piece of equipment that are really common in ornithological studies. They're these huge 6-meter-long nets with a really fine netting, and it's really hard for the birds to see them. And so what we do is we set up these nets in the grasslands where the birds live, and we might even put them around their nests. If we're trying to catch a specific bird, we'll play their playback. So we'll play a song, and they'll be like, who is that over there? I hear a new bird I haven't ever heard before. And they'll go over to investigate, not see the net, get caught in the net. And then we take them out of the net and are able to take all sorts of measurements on them, put the geolocators on them, and things like that.
And they're not harmed?
They're not harmed at all.
Not.
No, it's a little bit of a process detangling them from the net. Yeah. Imagine your wife's jewelry, necklaces get caught into a ball whenever they get all tangled, and you have to pick them apart, but then there's a bird in there too, and the bird's pecking at you. So, it's always a little bit of a process, but it's a lot of fun.
Now, my understanding is this summer you'll have a chance to do some of the actual netting yourself?
Yeah, I was there last summer as well, but this will be my second season up in Vermont, in Burlington, and our research sites are at Shelburne Farms, down in Shelburne, Vermont. And I will be leading a crew of undergrads with NOAA, and we will be looking for nests, catching new birds, and hopefully putting out 30 new geolocators to see who comes back again next year.
That's fantastic. So truth be told, I've seen that work in action. In my very first semester at UNE, Noah Perlett had invited me out to the field on campus, and I was able to see the students who had actually just caught the birds. I was blown away.
It's so much fun.
And they're so delicate, and the students were so careful. And these weren't graduate students, these were undergraduate students who were actually handling these birds. And I'd never seen anything like that in action. It was really awesome. And then to see how that simple class assignment translates in this kind of research is really just remarkable.
Yeah, it's such amazing opportunities that the students have here.
Yeah, that's great. Good luck this summer.
Thank you, I appreciate it.
look forward to seeing how this unfolds. And I think we're going to bring Amy back in here to get us on our tour.
Thank you.
All right, thanks so much, Katie. Dean Millen, I want to introduce you to today's guest speaker. This is Brandon Hotham.
Hey, very nice to meet you. Welcome back to campus.
Just arrived. So for those of you who don't know, Brandon was a double major in psychology and English, graduated in 2012, then continued on and did his master's in public health in 2014, correct? And did a lot of different research projects while he was here. I'm going to hand you the microphone because I see what they're doing. They're saying you don't have a... But, we were just going to talk about the benefits of undergraduate research, but who better to speak to that than one of our alumni? So.
Yeah, incredible benefit. I think so much of my research experience at UNE fuels what I do today professionally, right? So in my current role professionally, I work as the Chief Experience Officer for the Dempsey Center, which is a cancer support center that provides essentially every wraparound service for cancer support aside from traditional oncology medical treatment. So counseling, support groups, integrative therapies, and various other educational programs. And we use research to inform what we provide, right? We're a community-funded nonprofit. We have limited resources, so we want to go to the evidence and go to what's out there in the literature to determine what we offer to our clients in terms of impact. So I think research is just so critical personally and professionally too. When you think about even today, navigating what's out there for information and trusting what is right versus what maybe isn't accurate, my foundation in research here has really sort of served me both personally and professionally.
That's fantastic. Do I have time for a quick question? So when you came to UNE as a first-year student,
Yeah.
could you have imagined the job that you have now? Did you see that as a very obvious pathway?
Not even.
Tell me a little bit about that.
Yeah, so I mean, I entered into UNE as a biochemistry and pre-pharmacy major. I thought I would become a pharmacist. I think like many 18-year-olds, I thought I knew what I wanted, and then I discovered that that wasn't the best fit for me. But I think, throughout UNE, I made several shifts and changes. For a while, I thought I would go into clinical psychology. Then I thought I would go into law school. And finally, I did a research experience with a psychology professor who was doing public health-related work, and that's what led me to my public health degree. And then that's what led me into healthcare and public health in general. And so, yeah, I mean, I view it as like I was taking steps along the way, but I didn't necessarily know where the steps were leading. And it's a varied path.
I love that. Everybody who's watching out there who may not yet be in college, listen to this story. Come to college open-minded. Be ready to find a path that you may never even knew existed, and change your major if you need to or if you want to.
Absolutely.
I love this story. This is a story for liberal arts education in action. Outstanding.
Yeah, absolutely. Yeah, thank you.
Thank you, Brandon. We'll touch base when this is
we'll look
over.
forward to
Thanks.
seeing
Yeah.
you.
Thank you.
Yeah. Wow, that was so much better than what I was going to say, which was talking about critical thinking skills and problem-solving skills and the ability to formulate a research question and
That's the message.
Absolutely.
That was the message.
Absolutely. Yeah. And our next presenter, he has had internships, and he will be continuing on here in our Office of Research and Scholarship. And I think he would probably say the same thing, that these fundamental research experiences, even though it might not have been the path that he had thought of, will really serve him well in whatever he does next.
And when you say continuing here, he's going to become an employee of ours, a colleague of ours.
Correct. He's going to be working in our zebrafish facility. So we heard from Sarah, who was working with zebrafish. Brian, who we're going to meet as we deke around this corner, will be helping to support the zebrafish facility, which is perfect because he is a marine biology major and graduating in just 2 weeks. 2 weeks from today.
But sticking around.
Absolutely. So we'll be happy to have him again. So I'd like to introduce Brian Will, who is graduating marine science, marine biology major. And Brian's going to tell us about a project that he did in one of his marine affairs classes on aquaculture policy and management. So this is another example of integrating research and scholarship into a course. And he's going to be telling us about seaweed aquaculture policy and regulation in the U.S. So welcome, Brian.
Thank you. So my project focused on seaweed aquaculture here in the U.S. And as such an emergent industry, Maine has really taken a lead in the charge of growing out that industry. We have over 30 commercial farm sites here along the coast, and they are generating about $20 million every year. So here in Maine, the Department of Marine Resources is our state lead agency, and they have a very comprehensive and streamlined process allowing for easy access into the industry and allowing for it to grow. But there's a lack of federal policy structure that is hindering the industry from really developing across the nation and limiting it from its fullest potential.
Yes, we've heard a lot about food-based seafood tape. We've learned about lobster and scallops. Now we're learning a little bit about seaweed. So first, just a very basic question, and I should know the answer to this. Kelp and seaweed, synonymous? Is there a different?
Yeah, so seaweed is kind of the broad overarching term, and then kelp is more specified into different species and things like that.
And as far as the value of the seaweed, of the kelp, is it mostly food-based? Because I also see you have on here personal care products and others. I hear mostly about the aquaculture in terms of the food products, but I don't hear as much about other marketability.
Yeah, so Maine is really unique in that it's creating a market as well as looking at best management practices. I think when we think of seaweed and kelp, we think of sushi, but Maine is not really trying to break into that industry as it's too difficult. So they're developing these consumer-ready products, things like seaweed salad or dried chips and things like that. but also personal care products like soaps and lotions, and even looking at fertilizers for our plants.
And if I understand correctly, Brian, this started with a class project as a senior capstone?
Yeah, so this is a project in Susan Farady's U.S. policy in aquaculture management. So it's a new class this semester, and I think it's gone really well.
Outstanding. So I have to ask you 2 personal questions. The first is about the shirt. You chose it specifically for today, I'm guessing.
Yes.
Outstanding. Can you zoom in on that for us? We love our visual aids. And secondly, I have to ask you, is there any chance that your mom might be watching right now?
I think she might be.
And your mom is Janet, is that
Yes.
right? Well, Janet, if you're out there, we spoke on the phone maybe 2 weeks ago, and I don't think I got to say hello to you when you were on campus last time. So I want to make sure I get to say hi to you this morning. You got to be very proud of this guy. He did a great job. Brian, that's great. And you'll be sticking around UNE?
I will, yes.
And you'll keep me posted on how this all unfolds going
Yes.
forward. That's fantastic. All right. So, as usual, my colleague and co-host is off being distracted by somebody else, but that's okay. I'm going to grab the mic from you, and I'm going to take the lead, and we're going to start moving to our next poster,
All right.
which is right behind us.
Which is right behind us. And you know, Jonathan, as we've been talking, we've been seeing work that's been done in the classroom. We've been seeing work that's been done in research labs. Of course, that's not only the only way we can engage and work here at UNE. We have, of course, our 363 acres across the road. We've got our own island and research vessels, and so we carry out work in all of those ways. And in studio exhibits. Of course, we're talking about research, scholarship, and creative activity. So archival work that might lead to...
I just have to interject. We had an amazing art show on campus last month that featured all undergraduate student work. Some were art majors, some were not. The art faculty were phenomenal in their mentorship, and this was something to see. I really had no idea what to expect. But just like our science majors who are knocking it out of the park, our artists are creating world-class creative work. It's just an amazing place to be.
Yeah, we have great students here at UNE.
We have amazing students
well-rounded,
here at UNE.
absolutely. Arts and sciences.
Speaking of amazing students.
Speaking of amazing students, and speaking of ways to do research, we're going to see a project where the community can get involved with citizen science. So we're going to go over here to Heather Marvin, who is a first-year animal behavior student. And we also have Aaliyah Walker-Pesco, who is a junior animal behavior student. And they're going to tell us about the Maine Owl Pellet Project, which if you have any interest, pay attention, because I'm sure you're going to learn how you can be involved. All right? So Heather, welcome.
Thank you. So we're the Maine Owl Pellet Project, and so we're actually a collaboration between UNE, the Maine Department of Inland Fisheries and Wildlife, and the U.S. Fish and Wildlife Service. So we aim to engage the public and promote crowdsourced science to better understand the diet of Maine's owls. And this includes the threatened northern bog lemming, which we'll get into a little bit later. And we want to raise awareness about the importance of understanding the ecology and conservation of owls in Maine. And so as for describing owl food habits, this research actually hasn't been done in around 80 years. So it's very important that we get an updated census on their habitat and their food habits. And we want to detect the state-threatened northern bog lemming because this is where their habitat should be. However, whenever we go out to look for them, they like to evade our survey methods. So by using the owls, we are able to see whether or not they have picked up a northern bog lemming as a snack. And for our methods here, we get the pellets and they are heated to get rid of any bacteria, and then we dissect them to look for the jaw bones. And in the future, we will be doing DNA metabarcoding next fall.
So this is just some of our preliminary results as the project is still ongoing. So the project started in January of 2023 with dissections beginning in March. So we have received 15 total pellets to date, and in total we have received 28 prey items with an average number of 2.5 prey items per pellet. So the majority of the prey items that we have found is Microtus pennsylvanicus, which is the meadow vole, with Blurina brevicata coming in second, which is the short-tailed shrew. So if you look at this right here, these are just some instruction on how to collect the pellets. So when owls aren't out hunting, they actually nest in a roost. And this can be in tree cavities, on a branch, or just in man-made structures. And one way to identify the presence of owls is through whitewash, which is owl excrement. So this actually can resemble pine resin, but pine resin is sticky and fragrant. So we ask that whenever you see whitewash, odds are you'll also find an owl pellet nearby. So we just want to ask community members, that whenever they're out on a walk or a hike, or even if they have a tree in their backyard that they have seen an owl there, that they look for owl pellets so we are able to analyze them. And we are currently funded by the Maine Outdoor Heritage Fund.
That's outstanding. Thank you both so much. I have just a couple of questions. First, Heather, you're a first-year student, is that
Yes.
correct? And you're working with Dr. Olson on this project.
Correct.
So how does a first-year student get involved in a faculty member's research lab?
Well, I was actually very interested from the beginning, and I spoke to Dr. Olson about some opportunities that could be up and coming, and I did that last semester, and he said that there could be a project coming up in the spring, and when that time came, I got an email to be able to apply for this position, and luckily enough that I got it.
That's fantastic. And so you're an animal behavior major, and that's how you got to know Dr. Olson?
Yes.
That's wonderful. And finishing your first year, is this a project that you see continuing over your next 3 years?
100%, yeah.
That's wonderful. Thanks so much. I have a question for you as well.
Yes.
Now, I'm going to admit my ignorance as I ask you this question, because when I first learned about Dr. Olson's project, I didn't know about pellets. I didn't know where they came from in terms of the owl, and I made a misunderstanding. They come from the mouth, is that right?
Yes.
The pellets. And so some people may not know that, and it may help to get participation if you tell us a little bit about where the pellets come from.
Yes, so the pellets, when an owl eats a mammal or any prey item, the undigestible parts of that animal, they are formed into an oval-shaped mass. So this can be bones and fur, and it actually stays in their gullet for approximately 12 hours before it's regurgitated. So it's basically whatever they can't eat, they just throw back up.
So to be clear, folks, if you want to participate, we're not asking you to pick up poop.
No.
We're picking up regurgitated pellets.
Yes.
Yeah, that's
We
awesome.
do ask that if you are planning on collecting pellets, please bring a Ziploc bag because these pellets will be full of bacteria. So just to stay safe, please do not touch it with your bare hands.
That's great. And you're a junior rising senior, is that
Yes.
correct? And will you be staying on in this project as well?
Absolutely.
That's wonderful. That's great. Yeah, this is a great example of research and how the community can really get involved
Yes.
in helping us to do our work. So that's great. There you are. I thought you
Hi.
ditched me again. Thank you both so much. That's great.
No, I'm just in the back lurking and listening. Learning.
You're a good
Learning.
lurker.
I am a good lurker and learner. So I really like this project, and one of the things I like about it is that it's a collaboration and in partnership with some federal and state agencies. And
Right, and Dr. Olson actually will be on a sabbatical research leave in the fall out in the field collecting a lot of data for this project. So
lots
it's
of
great.
ways for people to be involved.
Yep.
So yeah, absolutely. And and speaking of federal and state agencies, we're really fortunate to have a lot of the projects here today funded by local and state agencies, but that's not the only way. We also have a number of private donors who give to specific disciplines or to sponsor students through our SURE program, for example.
And internal aid as well. I mean, we really invest in the work of our students and our faculty, and we're seeing the results of all of that investment today.
Absolutely, absolutely. So with further ado, this is one of our donor-funded students in the area of neuroscience, which is funded from the Kahn Family Foundation. And we're going to speak with Aiden Fox. And Aiden, you are a junior neuroscience student, and you've been working under the direction of Michael Berman in the School of Social and Behavioral Sciences as part of the Berman Collaborative. And you're going to tell us about blocking changes in DNA methylation of amygdalar CRF cells to reverse pain susceptibility attributed to the neonatal intensive care unit.
Have I told you how glad I am that you are here to do that part of this presentation?
I just show up to talk about the science words, really. So, Aiden, it's Dean Millen, and welcome.
Thank you so much for having me. I'm really excited to be on this Facebook Live. So as you were saying, my name is Aiden Fox. I've been a part of the Berman Collaborative for about a year and a half now. So basically, the Berman Collaborative overall is super interested in neonatal pain and how that affects later in life experience. So we do that by looking. We have a NICU simulation, so the neonatal intensive care unit. And then we look at how pain changes later in life because of that NICU. This is definitely something that hits home for me and for many people because a lot of people experience the NICU early in life. And of course we want to study how that affects later in life. So that's kind of what Dr. Berman looks at in general. So Dr. Berman has actually published multiple times on the idea that animal rodents that have been in the NICU simulation have chronic pain later in life and have a higher probability of experiencing that chronic pain. And so my project aims to to look at the mechanisms involved in that pain, to look at the neurobiological processes and of course DNA processes involved in that. And so as I'm sure you guys know, whenever you have an experience, it always is going to change your DNA. And so that basic idea is called epigenetics. And so we look specifically at a smaller part of epigenetics called methylation, and DNA methylation in particular. So basically, our animal model experiences our NICU, and then we inject a drug to hopefully reverse that pain that we've caused later in life. And so that's what we're doing so far, and we've actually thus far reversed it, hoping that we've determined the mechanism involved in pain regulation later in life.
Well, this sounds both intense and so important, right?
Yeah.
The applicability of this kind of research is without question for anyone who's a parent and is, as you said,
of course,
had a child in NICU and whatnot. How much time on the average week do you spend working with Dr. Berman in the lab? I mean, it sounds like this is really intense work.
I mean, so this is super intense work. There are about 4 projects going on at any given time. So I'm the lead of this project. I personally spend about 25 hours in the lab. Like I said, we use a rat model, so whether that be helping with husbandry or specifically working with my behavior test. So I would say about 20 to 25 hours. It is quite a lot of work, frankly.
And so for anybody who's watching who might be wondering how they get involved, I presume to be a project lead, you have to go through some training, you have to be taught certain techniques and responsibility compliance issues?
Yeah, so of course we go through and you have to, like you were saying, we have to get tested in it, and then you just have to prove yourself in the lab. So whether that's just like being there. That's kind of how I became a project lead. I was just trying to get involved in as much as possible. I joined the lab and I just soaked in as much as I could. And Dr. Berman kind of saw that in me, hopefully, and I think that's why I'm a project lead. So I've actually been doing this project for about a year now, and I joined the lab about a year and a half ago. So I was a project lead pretty fast within that time.
And so if somebody else is watching and they were saying to themselves, I would love to get involved in that kind of work, how do they get involved in the lab?
So on campus, you're always going to see posters saying, oh, join this lab, join this lab. If you don't see those posters, what you can do is you can just reach out to the PIs. So there are dozens of PIs on campus. Just send them an email and say, hey, I'm really interested in doing research. I want to expand my knowledge on what you're talking about and researching. And usually they're super receptive. I mean, I sent that email out to about 3 neuroscience PIs, and all of them got back to me, and I had interviews within the week.
And I presume you're a neuroscience major from that?
Yes,
That's outstanding.
Yeah.
Outstanding. Well, thank you so much for taking the time with us. This is really fascinating and important work. Absolutely.
Thanks so much, Aiden.
Wow.
I know. Right?
That's intense to think about how a project on campus working with rodents could actually change the way that children, infants, experience and deal with pain. I mean, that's remarkable.
And when I had my child just a few years ago, and I remember them taking him away to do some of these things and thinking about this work that's going on and how I could relate to that. So, knowing is power.
Yeah, and it really gives you a sense of scope when you think about the projects that we've seen so far and the range of topics that they're exploring, each one fascinating in its own right, and you realize just how complex the College of Arts and Sciences here at UNE. Yeah.
Absolutely. And one of the things when I think about the projects that we're seeing here today, so many of these students started in their first year, and then we sent them home when COVID arrived on our doorsteps. And and then a lot of these students started in their first year when we were practicing all those protocols of social distancing and masking and how do we even do research under these sort of new conditions in this new environment. And that didn't stop our students and that didn't stop our faculty. In fact, in many cases, including the next one we'll see, it provided some new research questions and new opportunities to explore this new reality that we have. So
well, that's a perfect segue. I don't need to add anything to that. Let's get right to it.
All right, and we're down to one microphone. No, we don't have 2 tables, but we do have 1 microphone. All right, so let's come on over here. We've got 2 psychology students. We have, I'll just stand here. A little bit awkward, but that's okay. We have Lauren Alley. Lauren, you are a junior psychology major, and Emily Newbra, who is a senior psychology major, both working under the direction of Dr. Jennifer Stigler-Balfour in our School of Social and Behavioral Sciences. And wow, I'm not a psychologist, so I'm going to hand off this microphone. Lauren, you're going to speak first?
All right, so thank you so much. So we're going to be presenting on our poster, Does Feeling Connected Matter? The Mediating Effect of Social Connection on Cognitive Load and Remote Learning. So we know that COVID-19 altered teaching and learning for a lot of people. Pandemic-related stressors and the shift to online courses has the ability to affect many aspects of student life. And research on the best practices for online learning focuses on how to balance our social connectedness as well as the strains on our cognitive load. So given the potential to go remote again, it's relevant to examine the optimal online learning environment to facilitate student learning. So we hope to examine the relationship between key psychological and cognitive factors and the ability to retain information from a recorded lecture, as well as whether the inclusion of a video of a professor will help or hinder our learning, and whether feeling more socially connected to the instructor would offset strains on our cognitive load and facilitate learning. So for our methodology, our participants included undergraduate students from the University of New England who were assigned to complete the task at the time and location of their choice. They were asked to watch a 10 to an 11.5-minute video in one of our 3 randomly assigned conditions. Condition 1 was only narrated slides where they could never see the instructor, just hear her. Condition 2 was picture-in-picture with the professor visible in the right-hand corner the entire time. And condition 3 was a full-screen video, intro and outro from the professor with narrated slides throughout the middle. After the lecture, participants answered a variety of questions, including questions assessing their retention and factors influencing their retention. And finally, they were asked to answer a variety of demographic questions.
So we performed a multiple regression analysis of our results, and they did find a bunch of positive and negative associations. But what does that all mean? So what we found was that presentation condition did not have a significant direct effect on recognition score. However, it did have a significant indirect effect. So our participants who scored higher on cognitive load performed significantly worse on the recognition test than those who had scored lower on the cognitive load. Moreover, students who scored higher on perceived social presence was also a significant predictor of cognitive load, meaning that our students in our audio-only condition, they scored higher on perceived social presence, lowering their cognitive load, offsetting the constraints of cognitive resources, and facilitating learning. And thus they were able to score higher on our recognition score. So although there was not a direct effect of presentation condition on recognition score, there was a significant indirect effect with social presence and cognitive load mediating the impact of our presentation condition. So by better understanding the variables and how they impact the negative impact they have on learning, we can hope to optimize and facilitate learning for students in the future.
That's wonderful. Thank you both so much. So I've got to ask you, because I was here as you were during the pandemic, and like so many colleges and universities at UNE, we very quickly pivoted to online remote learning in March of 2020 with very little preparation in terms of planning to do so.
Yeah.
And we did it, and I think we did quite well, all things considered. But now we're wrestling with the question, do we still do it? So what is the role of online learning now that we don't have to do it? But certainly there are some benefits. And I know this is an early stage study, so to speak, but are you able to extrapolate from this, what your thoughts are on balancing online with face-to-face learning?
Yeah, so we kind of found that, so one of our conditions that we investigated was the picture-in-picture version, which, as I said earlier, the best practices for online learning environments kind of focus on how to balance our social connectedness and our cognitive load, which I think is also something that's important in the classroom. So I think we learned, because we still use a lot of these methods today, I think it's still very important that we can manage our social connectedness as well as figuring out how to balance those strains on our cognitive load.
So what I hear you saying is, as long as there's a balance, it
Yes.
doesn't have to be all one or the other,
Yes.
but being mindful of that connectivity. Is that fair to say?
Yes.
Wonderful. And I've got a question, Emily, for you as well.
Yeah.
So now that this study is at this stage, what would you do next? I mean, if you needed to continue this line of research, would it be a different variable as opposed to picture-in-picture, or where do you take the research in the next direction?
So in future research, we would be interested in having participants who are previously aware of who the professor is. So for the majority of our participants, they are first-year students, so they don't actually know Dr. Seaweed, who is the presenter in these videos.
Right. So what we're outing her, right? That,
Yeah,
that the professor is also in the research, right?
Just a little bit. So there is a familiarity effect. So for those students, they don't actually know the professor, so it might explain the reason why the audio-only condition was better. So in future research, we would want to choose professors who have already a connection with the students, as well as choosing different kinds of modes of presentation of lectures.
Yeah, that's a really fascinating question because so in the spring of 20, when you went remote,
yes,
you already knew your professors.
Exactly.
And so would it have been different had you not known them from the start? Fascinating question.
No
Great.
problem.
I love it. I love
Thank
it.
you.
Yeah. Thinking on their feet. Amy, did you see that? Thinking on their feet.
You know, that is, what else could we expect from a graduate of the College of Arts and Sciences?
Absolutely.
You know?
Absolutely.
Yeah, absolutely.
As we both are, graduates of colleges of arts and sciences.
We really are. Yep, absolutely. So that's it. 8 posters, all
when you
different.
Say that's it. I mean,
no, that is not it. I mean, I don't.
Right. That may
Lee,
be
you
it
want to
for
do
us.
a pan?
That
Yeah.
may be it for us at the moment.
We've got close
But
to
if you look
70
around this
posters in
room,
here, more than 70.
We could have
Yeah.
spent literally all day
Absolutely.
interviewing students and learning about so many projects across the disciplines, across the arts and sciences.
Mm-hmm.
Our students, our faculty are just simply the best. And I think we don't need to say it. We don't need to
We
prove
don't need
it.
to. We
Our
brought them
student-
here.
Amen.
Absolutely.
All right, take us home.
All right, so we're going to conclude our Facebook Live session here. We've got lots more for the day. We are going to have lunch. We met our guest speaker, our alumnus speaker. We've got oral presentations in the afternoon. Again, more of the same. And then, I don't know, what follows that? A nap?
A nap, and then if you liked what you saw,
Yep.
and I know you did, same time next year,
next
we'll
year.
be right back at it. Bye,
Absolutely.
folks. Thanks for checking in with us.
Thank you. Bye-bye.
Have a great day.